Hello everyone. So do you have any problem in the last session? Can you hear me guys?
Yes, ma'am.
So please open this gene expression file. So now in today's session, we will see how to sort the data according to the significant genes. Okay. So this is our data, but we cannot say like the entire transcript is expressing, right? So can anyone tell me how to sort for the significant genes?
We can solve for the significant gene.
No, we didn't. You can see total entries is 1590,
Yes.
right? But we have discussed like in the last session also the p-value, it should be lesser than 0.05,
0.05, yes.
right? If it is okay, then the data is fine, right? But if
Yes.
the data is noticed, that means enough alignment is not has been done here, right? And also for the significant genes, we have to apply the color filter. So just select this lock to fold change.
Yeah.
Then there is a option of the conditional formatting. Just select that. Then go to new rule. Then make this format style into 3 color scale. Then what is the cutoff for the minimum value?
0.05.
No, Payukta. Like what is the cutoff for the down regulated and the up regulated genes?
1 or 2.
1 or 2? So
Ma'am,
for
upregulated, it's more than 1.
yes, correct. So this is the minimum, right? So minimum, you just change this lowest value to number here, and I will provide this cutoff as the -1. Whereas in the midpoint, you just change to number again, and 0 is for the no expression. And in the maximum column again, just change to number here, and now cutoff would be the 1 here, right? Clear till here? You also have to do along with me. So what I will do, I will highlight my upregulated genes with the red color. So change the color from here and make this minimum value, that is the green.
Ma'am, please repeat the options. We have to go conditional formatting,
Then go to the new rule.
new rule. Yes, ma'am.
Then you will see this small tab in the Excel, and just change this 2 color scale to 3 color scale, ma'am.
2 color scale to
3 color. You have to change from this drop down menu. Make it 3 here.
okay. Okay.
Done. Now you
Done,
can
ma'am.
see my screen. I have changed this minimum value, lowest value to number, because I'm putting the number here, right?
Right.
Just make it -1.
In the value we have to write -1.
Yes. Just see my screen, na? Like you just simply have to copy, like do the same thing.
Okay.
And change
Right.
the color of the minimum value. Make it to green. Done.
Yeah, done, ma'am.
Okay, now make it okay. Now you can see here, yellow color is indicating what? It is showing no expression. Whereas if you scroll down, you can see the green color, red color, right? So green color will indicate your down regulated one, and this red color will indicate the up regulated one. Okay. And another second filter you have to provide in the p value, because p value cutoff should be what?
0.05.
Yeah, less than 0.05. Okay. So just select the p value column, and there is a option sort and filter. Go to the filter option. Just select that. Now you can see the drop down menu again. Just select that. Go to number filters. Then make it to less than equal to, and just write here 0.05 and make it okay.
Ma'am, kindly repeat this
Yes,
p-value
yes.
process.
Yeah. Just select this p value, and there is a option, ma'am, sort and filter. Can you see?
Yes, ma'am.
Just go to filter there, and you will see the drop down menu here in the p value column.
Yes, ma'am.
Just select that, and then you will see the number filter option.
Yes, ma'am.
Then make it to less than or equal to.
Yes.
Done. Just write
Yes.
here 0.05
Okay.
and make it okay. Done till here, everyone? Now what you have to do?
1 minute wait, ma'am. My Excel has got stuck.
Okay. Who's this? Payukta?
No, Priyadarshini, ma'am.
Okay, Priyadarshini. Shall we move forward, Priyadarshini?
Ma'am, where to go and change sort and filter?
Which filter? Did you
Uh,
provide a filter to log to fold change?
yeah.
Then,
After I have selected the p-value,
okay.
open the sort and filter.
No. Yeah, sort and filter. Then go to number and filter.
Mm, sort smallest to larger.
No. No. Just go to the home, and in the home, right hand side, you will find sort and filter.
Ah,
Right? Just select that.
yeah,
Selected.
yeah.
You don't have to sort smallest to largest. There is a filter option. Can you see in my screen?
Yeah, ma'am.
Then provide the filter.
Okay.
Just select that number filter less than equal to. Done. Okay. Now what you guys have to do, just simply
I'm
copy.
not getting that thing done. Actually, I'm giving the filter, but it's not coming.
Okay, just wait. I will see your screen. Okay. Meanwhile, let me just copy this data.
In number filters, ma'am, what to do?
Yes. Number filter less than equal to
Okay.
0.05. Now, simply copy this entire data. and paste in the another sheet and just rename the sheet name that is to significant genes. Now, can you tell me how many significant genes did you got? How many? Okay. So let's see how many you are getting. So there is total 100. Why 100? You can see on the first row, there is a column like name we have provided, right? So we are not going to include that. So total 100 entries you are getting for the significant genes. Clear? Yeah, Khadija, Vishu, right? Imran, why 99 you are getting?
Ma'am, the first row is actually the gene locus samples, I mean the headings.
Okay,
So,
okay. Fine.
so we have 99. Yeah.
Now go to main file, that is the gene expression file. You have to keep the real data also, right? So again, go to sort and filter and remove that. Now in the significant genes, what you're getting here, you can see the status. In the status, all the entries are okay. Can you see that now? It has sorted according to the significant genes now. Done? Till here? Priyadarshini,
Yes,
do
ma'am.
you have the problem again? In
No,
this?
ma'am. I have done what you said.
Okay, fine. Now, second step is to do the annotation part. So what is the annotation, guys?
Ma'am, I have a question.
Who's this?
Imran.
Yeah.
Ma'am, I just checking the data. I mean, we don't have zero now. I
Which
think so.
zero?
I mean, the expression, just the positive and the negative values, right?
What are the
In
significant genes, Imran?
sorry, ma'am, I didn't get it. What you want to ask me?
See, here in the gene expression files,
Yes.
when I was providing the color filter here, so
Yes,
just
yes.
want to know which gene is showing the expression and which gene is showing no expression. And yellow color is indicating that this data is not expressing. Okay.
Okay.
Now, further significant genes are what? Significant genes are those that genes which are expressing, right? It can be the down regulated one and it can be the up regulated one, right?
Yes.
So on the basis of p value, what I did, I have provided the cutoff, right? That is the below than
Okay.
0.05. Why? Because if you are providing cutoff as the 0.05, it will just give you the only the significant genes, not the genes which is showing no expression. That is why the yellow color is not here.
For example, this is for the sample 1. If we have a compare with the sample 2 and sample 3 and in the same, I mean the same file, but
Yeah.
in some cases the gene is 0 and in the other samples, maybe it has been expressed. So on that cases, we will get the same results or we will have a different, I mean, just
No,
like
sir.
if sample.
Actually, what happened, like when you are having lot of samples, so
Yeah.
individually we are not dealing. Okay.
Yes.
So what we are doing, we are taking the average of all the control sample and average of all the treated sample.
Okay.
Okay, so then the tool itself, it is generating the log2 fold values there. Even
Okay.
like in the microarray also, the same thing we are doing with the help of our programming. So where we
Mhm.
are making the average of all the control sample and the treated sample. Okay.
Okay.
Yeah.
Got it. Thank you.
Yeah. So now what is the annotation part? What do you mean about the annotation, Shubham?
Labeling features.
That's
Huh?
getting the gene name using the gene ID.
Exactly. So in the Excel sheet, you can see there is, like, if I ask you what is the gene name or any other additional information, if I ask you, you are not getting here in the Excel sheet, right? So what we have to do, simply copy this A column. Make sure it is copied from significant genes, because further we are going to see the analysis for only for the significant genes. Just paste in another sheet. Now remove this gene word, delete this. So can you see in the NMIDs here, there is written .3, .2, .1. So these are different versions, right? So whenever like I'm putting this type of data in the UniProt, so it might happen, it won't take the NMIDs. So what you have to do, just select this A column, go to data, and there is an option text to column. Delimited, make it next, and select other. In the other, just put here dot and then finish. Done.
Can you kindly repeat the
Yeah. Did you copy this NMIDs in the next sheet?
Yeah, ma'am.
Now simply just copy this NMID, select this A column, go to data, there is an option text to column. In the text to column, there is an option delimited, select that and make it next, and then select the tab option and other. In the other, put dot there, then finish. Done.
Done, ma'am.
Now copy this NMID and go to browser and just type UniProt. Open. Now you can see like I am having the multiple transcript, right? So I want to retrieve all the multiple transcript together. Okay, I don't want to copy one transcript here and it will take lot of time, correct? So what you can do, there is a option ID mapping here. Just select that. And then paste all the NMIDs in this box.
Repeat, ma'am.
Did you open the UniProt?
Yes.
Then up, there is a, in the tab, blue tab, there is a option ID mapping. Can you see? Just select that.
Yeah.
copy all the NMID in this box. Now, there is a option from database. Just select that. And then move to sequence database. Select that. And in the sequence database, you have to select ref-seq nucleotide. Done. Mona, you just have to copy all the NMIDs in this box. Can you see my screen? Then below there is an option from database. Just select that. And in the from database, you will get the option sequence database. And in the sequence database, you will get the reference sequence nucleotide. And below you can see there is an option of map 100 IDs. So now you just select that.
Ma'am, I got 99, so I'm continuing with that.
Who's this?
Sumalata, ma'am. Sumalata.
Total 100 entries are there, ma'am, not 99.
Mine is 100.
Okay, now it is completed. Just open the file. Done. Now, can you tell me what is this 90 IDs were mapped to 104 result and 10 IDs were not mapped? What does it means?
Ma'am, 90 genes are mapped, but 10 genes are not matched with the reference gene.
Okay. Anybody else?
No results, ma'am, for me.
Who it is?
Sumalata, ma'am.
Can you just share your screen? Sanjana, there is a plus sign in the Excel sheet and you can create the new sheet there. Paste that. Pasted, ma'am? Yes, Shubham. Scroll up, it is already pasted, I guess. You can see scroll up. No, not from here, ma'am. In the box, there is a small slider there. Can you see on the right hand side? Yeah. Yeah, it is already pasted. So there is an option from database. Yeah. Just change to sequence database. And from the sequence database, there is an option ref-seq nucleotide. Can you see nucleotide? Yeah. Now map this ID. Now just open this file. Can you see the completed word? Open that. So you can see 90 IDs were mapped. Scroll up, ma'am. Scroll up, scroll up. You can see 90 ID were mapped, right? So there were total 99, right, in your file. So out of 99, 90 IDs were mapped here. So that means 90 IDs, for that you are getting information from the UniProt. But the 9 IDs were not mapped here in the UniProt. Okay. Clear? Now, if I want to know what is the biological information or maybe the cellular component, so we can also customize the table from the UniProt. So what you have to do, there is an option customize column up there. Can you see customize column? Can you see that? Yeah. So just wait. And also in the table, you can see only gene names, organism, length, they have provided, right? What if I don't want to deal with this? I just want only the genes and the functions. So what you have to do, go to customize column and you will get lot of option. Now, can you just select the name and taxonomy, ma'am? Yeah, so in the name and taxonomy, what I want, only the gene name. So just remove the entry name from there. Organism name I don't want. Proteins name also I don't want. Just keep only gene name, guys, in the name and taxonomy. Then go down to the sequence. Yeah. So in the sequence, scroll down. I don't want the length in my data. Just remove the length. Now you can see miscellaneous there option. Again, you can uncheck that. So it's up to you which type of data you want to work like in real time. You can select that, but for now we don't want, so that is why I'm just unchecking this. So now we will go to the gene ontology. Just select that, and from here just select the biological process, cellular and molecular. Can you just select that 3 option? Okay, now scroll, can you see the external resource below? Yeah. Now in the external link, you have to go to the, scroll down a little bit. Genome annotation. Just select that. So in the genome annotation, I want gene ID. Just select that. Gene ID. And KEG. KEG. Can you see the KEG word there? Yeah. So all of you have provided the filter here. Shall we proceed? Now save this file, ma'am. Now see, it has been updated. Can you see this table? There they are giving you the NMIDs, the entry gene names, gene ontology, biological process. Can you scroll towards right hand side, ma'am? Slider, there is a slider below. Scroll down. No, not from here, not from here, below. There is a slider. On the left hand side, can you see that? Yeah. No, below. Yeah, move towards right. Now see the page is updated to cellular component. Scroll down now again towards right. Molecular function you can see, right? So you can see like after putting all the filter to the columns, it has been updated. Can you see that? Now we have to download the file from the UniProt, correct? Because every time I don't want to go and run the UniProt. So for that, you can see there is a option download up there. Just select that download. Now, can you see the format that is in the FASTA, right? So just press the drop down menu. I want my sheet to be downloaded in the Excel format. Just download in the Excel. Now I don't want to compress my file. What you do, there is a option compress yes or no. Just select the no word from there and then download, ma'am. Same thing you have to do in your system, guys. And when it is done, let me know. And then from the downloads, you can keep that Excel file in the CuffDiff results. So everyone downloaded this UniProt data. You can answer me. Unmute yourself, guys.
Yes, ma'am.
Yes, ma'am.
Okay. Then open that Excel, which you have downloaded from the UniProt. Can you see my screen? So as Shubham said, so there are like chances of like you are getting the multiple entries. If you scroll down, can you see the total entries? 100 and like 14 entries you are getting, right? So that means this data might have the duplicates here, right? So what you have to do, just select the A column Then go to data. And there is an option of removing the duplicates. Just select that. Expand the selection. Remove duplicate. Unselect all. And just check the from, because column A name is the from, right? So just select that and make it okay. Done till here?
Ma'am, after remove duplicate, then
Did you selected remove duplicate? Can you see my screen? Just select the expand the selection.
yes, done.
Done. Okay. Now in this data, you can see the IDs you are getting: gene names, biological function, cellular function, and the molecular. And these are the KEGG IDs that we are going to use further in the KEGG database so that we can generate the pathways. So that is why like I have downloaded the KEGG from the Uniprot. Now just save this file. Done. Now what you have to do, just copy the entire data.
Ma'am, 1 minute, ma'am. In remove duplicates, what we must do, ma'am?
Okay, you can do. Meanwhile, just copy the data from the ID mapping and go to your main file, that is the significant genes here, and then paste the data.
Ma'am, I think the data didn't show any duplication.
No,
I don't
Imran,
know,
there is the duplication.
but I didn't get, I tried to three
Okay,
times.
what you can do, just copy that data and just paste there in this main Excel sheet where you have
All
created
the data.
significant gene. Okay, further it will be
All the
sorted. Yeah.
okay.
After Q value, just keep the K column as blank and put the data in the L from the L column, guys. Done. Now, next thing is, shall we proceed further?
Yes, ma'am.
Okay. Now, the data which you have taken from the Uniprot, just select the data, Control Shift right arrow, Control Shift down arrow. Then go to data. There is a option sort. Just select that. And now we are going to sort on the basis of this entry. So you can see there is a sort by entry. Just select that and make it to A to Z only. Then okay. So we have like sorted this data in the ascending order. So same thing you have to do for this also. Control Shift left arrow, Control Shift down. Just select the data, go to data, then sort. Now here we are going to sort on the basis of which? Uh, Sorry guys, we have to copy the entire data. I didn't selected this NMIDs. Sorry for that. You have to copy the entire data. You don't leave this A column. Now just sort this. Go to data, sort from, and make it okay. Same thing you have to do on the left hand side also. Data, sort on the basis of NMID. So that NMID name is the gene column. Just select that A to Z. Now we have to cross check whether this information like which we got from the UniProt, is it matching with this NMIDs or not, right? So for that, you have to put one formula in this K column. So just write here match. And put the formula equal to this L2, then equal to, just select the A2. Then enter.
Please, can you repeat that again? Please. Okay. Equal to L2, equal to 8. Okay.
Should I repeat, Jezreel?
Okay, I've seen it.
Make
Yes,
sure
ma'am, repeat it.
you have this data with you where you have like separated the version.
Yes.
Yes, just copy that first. NMIDs from here. Or from here also you can separate, because you can see the data which you got here from the UniProt. You are not getting with the version number, right?
Yes.
Can you see that? But this is the same ID. Did you see? So either you copy from that sheet or either you can separate here also. Just put the column here before. The data is copied. Copy this data. Move to new sheet. and remove the version number from here also. Then copy and move to gene, like significant gene, and paste here. Now we will see whether this data is matching or not. Just put here equal to L2 equal to this A2. Enter. And there is a autofill button. You just select that and you will get this type of data, true entries. So true will indicate that this NMID is matching with this, the data which you got from the CUFTIF, right? So we can say if it is true, that means this particular information which you have downloaded from the UniProt is right here. Now if you scroll down, can you see the false entries? Right? So false means this data is not matching. Can you see this 63 row? The ID is 748 and this is what 467, right? But if you see to the next 64 column, this ID is matching with this, correct? So wherever you are getting false entry, so for that what you can do, just cut this data and paste it below. Then delete this. Shift cell up. Okay. Now from here, true entry, just autofill your data again. Now next again, you're getting false. Why? Because the IDs are not matching again. Now you have to see which IDs is not matching here. 1157, right? So here it is 1537. Right? Are you getting it or not?
My data is not matched. False it is coming.
Who is this? Sumalta ma'am.
Yes, madam.
So every data is false.
Yes.
Show me. What about others? Are you getting
Ma'am.
true entries or false?
Ma'am, actually,
Getting false.
actually the problem is that I think the duplication was not removed when we were doing.
So you didn't remove the duplicates, you mean
I
to
think
say?
this is the problem. Yes.
Okay. One by one, whoever is getting the false entries, just share your screen, okay? Okay, meanwhile, you can see the Sumalta ma'am screen. Can you see that?
Yes, ma'am.
Okay. Can you just stretch ma'am this L column so that I can see the NMIDs? Just delete this ma'am.
Which L column, ma'am?
Yeah, this all the data which is
Oh.
like you have copied from the UniProt, ma'am. Delete this. Not this, entire. Yeah. Yes, delete that. Go back to your ID mapping. ID mapping sheet.
Excel, ma'am?
Yes, that Excel ID mapping, which you downloaded from Uniprot, ma'am. Open.
Mm.
Yes, Shubham. Right. Did you open, ma'am?
Yes, ma'am.
But I can't see. What about the others? They can see the Uniprot data?
Yes.
Okay. Ma'am, did you, how many entries are there in this, in your, this ID mapping?
99, ma'am.
Okay. So total 99 you are getting here. So what you can do, just select this A column. Yeah. Go to data. There is an option remove duplicates. Can you see that? Remove. Expand selection. Remove duplicate. Unselect all. Then select the from entry. From. Just select the from column. Make it okay.
Entry also, ma'am?
Oh, no. Only from.
Okay.
Make it okay. Okay. Now just
Because already I have done this.
okay. Now copy this, like I was checking whether like you have removed or not. Just copy the entire data now. Not like this. Okay. You can do like control shift right arrow also. Copied.
This one I'm unable to do this.
No. Go to first cell. First cell. Just select from. Just select from. Only from. Yeah. Just type control shift right arrow, ma'am. Not down arrow. Control shift right arrow first. Yeah. Not this. Yeah. Copy.
Yes.
Go to your main file.
Shall I paste here?
Actually, your screen is lagging in between. I don't know.
I pasted in the L column, ma'am.
Hello.
Hello?
Pasted.
In the L column, ma'am.
Okay, just wait. Let me see your screen. I can't.
Hello.
Hello, who's this?
Ma'am, Arpita ma'am, actually, ma'am, recording would be there for matlab after this, after 18th October.
Hello.
Yes, ma'am. Actually, after this session, matlab, after 18th, recording would be matlab, available after that.
Yes.
How much days it would be available?
No, ma'am. You can download in your system now.
Matlab, till how many days? Matlab, it would be after 18th.
No, like 18th is the last day, right?
Yes, actually, I was not getting time to complete.
Okay, so the recordings have been like given in the WhatsApp group, and also I have shared in the YouTube also and the Drive also. If you have the access, you can download from that.
But after 18th, how many days it would be available after 18th?
No, ma'am, that you can download. Like you have already the access. Like I have given the links. Right?
So after 18th, it would not be there in LMS.
It would be there, ma'am. It would be there.
Okay, well, 2-3 days it would be maybe.
No,
No,
ma'am.
no, you can
access
download
anytime.
that in
Yeah.
your computer. Then you can see.
In that too I understood, but after that 18th it would be available or not? That only I'm asking.
It will be available, ma'am.
हाँ मतलब वही 2-3 days it would be available. Okay.
Now,
Okay, it-
Sumalta ma'am,
and-
what you can do?
Yes.
Okay, and it is not tough, no, ma'am? Okay, thanks.
No. Whoever it is stuck with the annotation part, they can see her screen. And now try to sort this data, ma'am. Just copy,
Okay,
just select
thank you,
this
ma'am.
Uniprot data. Okay.
And thank you, ma'am, for this.
Yes.
And I pasted in the L column, ma'am.
Yeah,
In the
I
main
saw. You just select the from and the keg entry. Just select the data.
From to take. Okay.
Select the right hand side data. Complete.
Shall I go
Control
to
shift
down?
down. Yeah. Now go to data sort. There is a sort option. Can you see? Sort.
Yes.
Yeah.
Hmm.
Just select that. Now sort by from. Just select from. Make it okay. Done? Now
Yes.
same thing you have to do like on the left hand side. You have to sort according to the ascending order. Okay. I guess you have already sorted, right?
Yes.
Yes. So what you can see, like on the first A column, there are different version. Can you see? Dot two. We have already removed, right? When you were pasting in the Uniprot, right? So what you can do, just select the A column from here and then paste in the next sheet.
New sheet.
Yeah, new sheet. Done?
No, ma'am. Yes, ma'am.
Now remove this gene word. Now again select the A column.
Yes.
Go to data, then text to column. Yeah. Then again delimited next. Just select the next button. Then go to other and then put dot. Done, ma'am? Hello? Can you hear me?
Ma'am, can I share my screen to check the duplicate?
Yeah. Did you follow the same step, Imran?
Yes, ma'am. Actually,
okay.
I did, but
Let's see.
I cannot remove the duplicates.
Okay, just go to the ID mapping data.
This is
How many entries did you got this here?
115.
Okay, so scroll up. Okay, just select the A column.
Oh, okay.
Only A column.
Yes.
Now, there is an option remove duplicate. Can you see that? Next to text to column?
Yes.
Up. Yeah. Select this.
Okay.
Expand the selection, remove duplication.
Okay.
Then unselect all. Unselect all.
Oh.
There is an option up there. Unselect.
Excuse me, ma'am.
Yeah. And
Hello?
just select the from. Just wait, ma'am. Imran, just select from button and make it okay.
Oh, okay. Yeah,
Can you
24.
see?
Yeah, it's
Okay.
okay now, ma'am.
Okay.
I will proceed further. I know.
Keep your like screen share so that other can also follow. Okay, just wait.
Okay.
What,
Okay.
Arpita? Hello, Arpita.
Please, can you continue, ma'am?
Okay, we can continue. Yeah. So, can you maximize this, Imran, so that everyone can see what you're doing?
Okay, ma'am.
Hello?
Yes, Arpita.
Ma'am, actually YouTube link तो it's shared in.
मैम प्लीज आप ये जो क्वेरीज है प्लीज आप व्हाट्सएप में डाल दो सो दैट वी लाइक कैन नॉट डिस्टर्ब द सेशन वी आर नॉट एबल टू कंप्लीट द एनोटेशन पार्ट टिल नाउ
Okay, fine. But other thing I was asking that in that other link was also there, other medium apart
प्लीज
than
डोंट
that.
शेयर द लिंक हियर गाइस इन द Zoom.
Okay, ठीक
So
है.
like
नहीं,
whatever the
YouTube link तो
queries,
I got, but other link I also want. No, that's why I was asking other
it
thing.
will be, you can contact the HR ma'am please.
Okay, okay. Thank you, ma'am.
Amran, just wait, you don't have to put the formula here. Delete
Okay.
delete this first. So did you sorted the data?
Yeah, I've already sorted. I mean,
Okay, fine, fine, fine. Now put the formula here. M2 equal to A2. Yeah, enter. There's the autofill option. There's a plus sign, Imran. Don't do like this.
okay, ma'am.
I will tell you.
Mm-hmm.
Yeah, can you see plus? Double select, double click here.
Okay.
Yeah, fine. Now, wherever
Yeah.
you can see true entry, that is perfectly fine, right? So that means that IDs are matching and that is your information you are getting for that particular NMIDs. Okay. Now, the
Okay,
thing
ma'am.
is how to sort the false entry. Can you see the false entry here?
Yes, ma'am.
Right. So can you tell, just select this 63 row. Go up, go up, go up. 63 row, just select that. Can you tell me which ID is not matching from our data?
Ma'am. The
748.
Correct. 48 NMI ID. Can you see on the left hand side ID which
Yes,
is
ma'am.
ending with 748? So it is not
Yes,
matching,
ma'am.
correct? So what
Yes.
we can do here, just select this. Can you see the false up option? False word there.
Yes, ma'am.
Next
Yeah.
to that there is a like 0.0 something. Can you see the values? Yeah. Yeah, like
Okay.
0.028, right? Select that.
Yes, yes.
Select that. Now just press Control Shift left arrow.
Control Shift
Left
left
arrow.
arrow.
Now you have to cut this data, okay?
Okay, ma'am.
And paste
Cut.
it below. Go down.
Here.
Yes.
Oh,
What you did? No, you don't have to cut it like entire.
okay, okay.
Shall we do like in my systems? Otherwise, like you won't be able to.
Yes, yes, ma'am. Yes, yes. I will
Yes.
follow.
Yeah,
Thank you.
yeah.
Thank you.
Wherever like you guys are getting stuck, just pause me, okay? In between. So here you are getting false entry, right? So that means, not this. This one, right, Imran? So 748 is not matching with our data, right? But we cannot lose our data, correct? So what you can do, simply Control Shift left arrow, cut this and paste it below. Done?
Below.
Yeah.
Okay.
I will tell you why, okay? Now, this cell is free, right? It is not containing any information. So what you can do, just right click it and there is a option shift cell up. Can you see? Make it okay. Done? Done till here? Imran? Done? Hello?
Yes, ma'am, I have done.
Done? Now, can you see,
Yes.
like in the 63, it is matching 467, right?
Yes,
Now,
ma'am.
just select the true entries and autofill it again.
Okay.
Done? Done till here, guys? You can respond, please.
Yes, ma'am.
Okay. Now, next we have to sort this type of data. You can see again, like the data you are getting NM1537, right? But here you are getting 1157. Again, it is not matching. That is why it is showing false here, right? So we have to see which IDs are not matching. Can you see in the 71 row, the data is matching with this, right? So what you can do from the 65 till here, you have to cut this data. Did you selected this? From 65 to 70. Now again cut this because this data is not matching and paste it below. Now again the cells are free. What you do, just delete this. Delete, shift cell up and make it okay. Now again you have to go select true and autofill it. Now again see the IDs are matching. Can you see that? Done?
Yes, ma'am.
Now scroll down. Now can you see for your data total 91 entries, you got the complete information. Can you see that? And also when you were working in the here like in the UniProt, 10 IDs were not mapping, right? Now if you scroll down here in your file, so these IDs are not mapping. Okay, that is why like we are not getting the information from the UniProt. So sometime what happened, like still the research is going on. So that is why like why I have pasted it below, because we cannot lose the data, right? Sometime the databases are getting updated, right? So that time what you have to do, you have to go again to the NCBI just to know which like other information it is there or not. Okay, and you can complete your data. Clear?
Okay.
Clear, Jasre?
Yes, I think so.
Okay. Now, can you see these IDs are matching with this, right? So this is of no use again now, because the same IDs like you can see on the A column also. So what you can do, just delete this. And you can also delete this match. It is just for us only. So delete this. And also, like, if you want to keep this entry gene name, it's up to you, but I will not keep this entry. I will just keep the gene names, functions, and the gene ID and the KEG IDs. So this is how we do the annotation part. Did you got the complete information now? For this, if I ask you what is the gene for this particular NMIDs, you can say LCAT is the gene,
Correct. Okay.
right? And
Yes, ma'am.
if like somebody asks what is the function of that, you can see like this is the function. And also it's not about like every time like in the research you are dealing with only the function part. So in the UniProt there are a lot of option, right? So you can just select like what other information want, like if you want organism name, if you want sequences, so you can select. So you know, now you know already how to download this data, right? So our task is just to download from the UniProt. We downloaded and we mapped and we saw like we got the complete information. Okay. Now, next task is we have to sort the data according to the upregulated genes and the downregulated genes. Okay. So now can you tell me how to sort the upregulated and downregulated genes?
Ma'am, just to go to the log to fold and ascending, descending order,
Yes,
maybe.
Imran. Yes. So, like we have discussed already, like in the previous session, cutoff for the upregulated gene was more than 1, right? Equal to greater than 1. So, what you can do, just select this log to fold change value and provide the filter. Then go to number filter. There is a option greater than equal to. Make it 1 here and okay. Now, every time I cannot put the filter, I want to save this now. What you can do, just simply copy this data for the upregulated and paste in the next sheet and just type here upregulated genes. Now again go back to the significant genes, remove the filter. Now we have to sort the data according to the downregulated genes. So again put the filter, number filter, less than equal to, put here -1. So it will indicate only the downregulated genes. Make it okay. Then again copy. And paste in another sheet. And rename it as the downregulated genes. And make sure whenever like you don't want to lose the significant gene data also, right? So what you do, just remove the filter again. Now you are again back to this. Now it has been separated. You can see downregulated genes and upregulated genes. So if like somebody asks like from your data, how many genes were expressing the expression, like which are the significant genes? So you can see like total 100 genes. But out of 100 genes, which genes are the downregulated? You can see 7 entries are my downregulated. Whereas for the upregulated, 18 entries you got. Clear? The annotation and the sorting of downregulated and upregulated. Clear?
Ma'am, I have a question.
Yes.
How many total genes do you have on the Excel sheet now?
Like
Significant thing. I mean the total, the up and the down.
up and down. You can see
Yeah.
the downregulated data here.
No, I mean you have only 21 genes. The
No.
significant genes.
You're talking about significant genes?
Yes, significant.
No, total you are getting, how many entries you are getting total? You are getting this.
Yeah, I mean, we are getting like this. So we have only, I mean, 5 or 6 genes that are downregulated, and only 10 or 15 genes are upregulated. But I guess that if we select, I mean, the above, I mean, the positive, I mean, from the 0.01 and the minus 0.01, we can differentiate like this. I mean, the upregulated and the downregulated.
Yes, sir, I got you. So what happened, like whenever you are doing the project work, so
Yeah.
it is not sure the same cutoff you have to provide for the Excel sheet, sir. If I'm saying the cutoff is the more than 1 for the upregulated genes, so sometime what happen, like you are getting the data that is the 0.9, right?
Yeah.
Like the entries you are getting 0.9. Okay, so if I provide the cutoff that is the 1, I keep that standard cutoff as the 1, so that means there will be no genes, correct?
Yes.
So it totally depend like what type of data you are getting, and you have to see like in the log to fold change value, so which genes like showing the nearly like to which one, or maybe 0.9. If I can see if the data is near to 0.9, that time I will put the cutoff as the 0.9, not the 1, sir. It is depending on the
Okay.
data. Okay, but make
Yeah.
sure like for the significant genes, for the p value, always the value will be the 0.05, sir. Okay,
Yes, ma'am.
so not for the upregulated and the downregulated. Okay.
Sometimes it's a confusion that maybe the positive value is considered as the upregulated and the negative value is considered as the downregulated. Is it
Yes.
right?
Yeah, this is right, because we have discussed this log to fold change also, like in the last session, why we were saying the positive as the upregulated.
Right. Okay, ma'am. Thank you.
Yeah, when I was dealing with the some other data, that time I was putting the cutoff as the 0.8 there for the upregulated genes, not the 1, because I can see like in the Excel, like the clearly it was near to 0.8, so that time I put it like as a 0.8. So it's up to you, sir. Okay, if you don't want
Okay, ma'am.
not want to lose the data, so it's only like up to you which cutoff you are like providing for the upregulated and downregulated, but make sure p value should be less than equal to 0.05, sir. Clear?
Yes, ma'am. Clear.
Excuse me,
It's
ma'am.
fine.
Who's this?
Hello.
Yeah.
Ma'am, there is moderate genes also we have to follow, because orange color would be a moderate genes if I am correct.
Yeah. So that is what, see, the darker red is what it is showing higher expression, right?
So, we have to consider both three of the genes we have to consider, matlab, like
Yes.
that.
Yeah.
Nothing like that. Okay. And next thing, ma'am, ki for that significant upregulated and downregulated, we have to follow look for p value and log to fold change. Both we have to look,
No,
or
ma'am, we
only log to
already,
fold parameter.
if you saw in the gene expression, we already sorted on the basis of p value, right? So we have already
But we
did it
don't have
on-
to consider over here both parameters. I think so.
Yes. First, you
Only
have to sort the significant gene on the basis of
Okay.
p value.
if one is sorted first, so then we don't have to consider both. Otherwise, both is considered in this whatever upregulated and downregulated things.
Yes, ma'am. These two important. Like these are the important
Thank you,
parts.
ma'am.
Yeah. Okay. So any doubt in the annotation, or maybe the downregulated and upregulated? Because we have already stretched the session and we'll end here. I can't hear you, ma'am.
I think please, if the session will be pasted early.